DNA replication. This is a piece of DNA, aka deoxyribonucleic acid. It is made of tons of nucleotides. A nucleotide is made up of a deoxyribose, a phosphate ion, and a nitrogenous base. There are two groups of nitrogenous base, and each group has two bases. The purine base with a two-ring shape, adenine and guanine. The pyrimidine base with a one-ring shape, thymine and cytosine. In the double helix, the A and T bases with always and only bind to each other, which means the G and C bases are buddies. The bases are bounded by hydrogen bonds. The A and T bases are binded with 2 hydrogen bonds, and while the G and C are binded with 3. The two strands of DNA are antiparallel to each other. One strand has a direction of 5' to 3' and the other has direction of 3' to 5'. The 3' and 5' refers to the carbon counting starting from the oxygen of the deoxyribose. Now we can talk about how DNA molecules copy themselves. The replication of DNA starts at the origin, a specific spot of the long double strand of DNA. It denotes the replication forks, the area of active replication. Here is what happening at the forks. The helicase enzymes unzip the strands. But in the unzipping process, the rest of the DNA strand will receive tension. Sometimes the tension is too strong and will cause the DNA to be too tight to split. There comes the topoisomerase enzyme. There are two types of topoisomerase, one is type 1 and the other one is type 2. Type 1 breaks one side of the strand and glue it back to release tension. Type 2, aka DNA gyrase, does the similar job except it is cutting both of the strands. Once the two strands got separated, the single-strand binding proteins come to hold the strands and keep them stable. Once the fork is stable, DNA polymerase III starts putting new nucleotides to the two strands. The strand with direction 3' to 5' is called the leading strand. Since DNA polymerase III only works in the 5' to 3' direction, it can directly and continuously put the nucleotides onto the leading strand, before putting the nucleotides down continuously. It needs a single primer from the DNA primase to give it a start signal. The primers are small pieces of RNA. For the other one, the strand with direction 5' to 3', it is called the lagging strand. Due to the picky behavior of the DNA polymerase III, lagging strand needs the DNA primase to keep putting our pieces of RNA primer ahead of the 5' end of the lagging strand. Then DNA polymerase III adds short sequences of nucleotides to the primer to fill in the gap. Those nucleotides has a fancy name of Okazaki fragments. After the Okazaki fragments are completed, the DNA polymerase I will replace the primer with a DNA nucleotide, and the DNA ligase comes to ensure bonding between the fragments and the replaced nucleotides. Then the replication is completed. Woohoo!